In the Labster simulation, you will review different types of ELISAs and also learn how to set up and interpret a sandwich ELISA. So let me review what a sandwich ELISA is and how it works. Remember, in the sandwich ELISA, you have a well that's in a microtiter plate similar to this one. So this plastic plate is filled with all of these little plastic wells, and that's where you add the different reagents to. The first antibody that's added is called the primary antibody. So the primary antibody is going to bind to the well, and then you're going to add what's called a blocking reagent. This is going to bind to any of the spaces that are not covered with antibody, and this will prevent the other antibodies and antigens from sticking to the well non-specifically later. Then you're going to add the sample. In the Labster simulation, the antigen that you're trying to quantify is actually a protein, factor 9, which is used for treating people with hemophilia. And you're trying to figure out how much factor 9 is made by different cell types. And so you add the sample, and if factor 9 is present, then it will bind to the antibodies that are stuck here. And then you're going to wash the wells out. An unbound sample gets washed away. So anything that's not factor 9 should get washed out of the wells. In step 5, the next antibody is added. This is sometimes called a secondary antibody, and it's the one that has the enzyme conjugated to it. So you can see the purple thing here is the enzyme. And so this secondary antibody, if the factor 9 antigen is present, then the secondary antibody will bind to it. Then any unbound secondary antibody gets washed away during a wash step. And then the next step will be to add the substrate for that enzyme. The substrate is added, and if it's present, then the enzyme converts the substrate to the colored product. In the Labster, you'll also use sulfuric acid as a stop reagent to stop this reaction. So if you just want a qualitative yes or no answer, you would look for the presence or absence of color at the end of this ELISA. But what you're going to do in the Labster simulation is actually quantify the amount of factor 9 that is in the sample. You're going to do that using what is called a standard curve. A standard curve is when you use a known amount of the antigen you're trying to detect to create a dilution series that will give you a standard curve that you can compare the unknown samples to as a way of estimating the concentration of the antigen in the unknown samples. So this is what a standard curve might look like. You can see at the top here is the darkest color, that's the most concentrated. And then the reason it's decreasing as you move down the plate is because a serial dilution is performed, where you take this top well and you dilute it into the next well, and then you take the row B and dilute that into row C. You take row C and dilute that into row D. So if row A started with 200 microliters of volume to start with, then the way you would do the dilution is you'd add 100 microliters of buffer to row B, and then you'd take 100 microliters from row A and move that into row B. So row B now has half the concentration of row A. And then you'll do the same thing down the line. For row C, you'd add 100 microliters of buffer into all the wells in row C, and then add 100 microliters from row B into row C. And same thing for row D, row E, row F, row G, and row H, all the way down. So each time you move down the column, the concentration is cut in half. And you can see that reflected in the intensity of the color. Now in the Labster simulation, it's going to ask you this question. The first well contains 200 microliters of standard with a concentration of 90 nanograms per milliliter, although this is actually a typo. They meant to write microliter. You can see that here in the image, microliter. If we want to use a dilution with a 1 to 2 dilution factor, how much sample should be transferred from the first well to the second well that contains 100 microliters diluent? And so you can see that in the last slide I showed you. This is a 1 to 2 dilution. When you do 100 microliters buffer and 100 microliters of the previous concentration, then that's a 1 to 2 dilution, or 50% or half, or however you want to express it. You would take 100 microliters and add it to the 100 microliter of diluent. Diluent is just the buffer, the thing you're using to dilute the concentration. And then later it's going to ask you, well, what is the concentration of each of those rows in the standard curve? And so you can calculate that just by dividing by 2. It tells you it's 90 nanograms per microliter to start here in row A. And so when you move down to row B, that's just divided in 2. It's going to be 45. Row C will be 22.5 nanograms per microliter. Row D will be 11.25 nanograms per microliter, and so on and so forth. All right, so once you have the standard curve set up correctly like this, how is it used to estimate the concentration of an unknown sample? In order to do this, you have to graph the color versus the concentration. And so here you can see the graph that might be made from plotting the color measured as optical density versus the concentration. Generally, this kind of curve is expressed as a log. That means that this is log and this is log. And here's the formula for the line that's generated here. Log y, that's the y-axis, is equal to the slope, 0.998 in this case, times log x, which is the y-axis here, so that would be the concentration, minus 1.437. And remember, 1.437 is just where this line would cross the y-axis. And when a standard curve is plotted, the r squared value here will tell us how good a fit is. How well does a straight line fit these data points? And the closer to 1, the better the fit. So 0.99 is a pretty good fit here. And then what the Labster simulation is going to do is it's going to ask you to calculate the concentration based on the optical density, right? And so if you have a sample with some unknown amount of the protein you're trying to measure, then you would measure the optical density. Let's say it was 0.5. And so wherever that 0.5 hits the line, that's about what the concentration would be. So it looks like it's maybe around 1 or so, 1 nanograms per milliliter. And so you can use this formula to calculate the concentration based on the measured optical density. And I've written that out for you here. So here's the formula. And you can rearrange the formula where you add 1.437 to both sides and then divide by 0.9988. And this isolates log x on one side. And then if you just plug in the number for log y, then you can solve the math and to figure out what log x would be and solve for what the concentration of the unknown sample is. For example, if the optical density measured for the well with the unknown sample in it was 1.8, then you could just plug that in for log y. 1.8 plus 1.437 divided by 0.9988, and that would give you the answer of 3.24 nanograms per microliter as the concentration of the unknown amount of protein. Hopefully this will give you the information you need in order to successfully complete the ELISA Labster simulation. Let me know if you have any questions.